So it's been a side project in my lab, and it's work that's been done mostly by undergrad students, and we've gotten some nice results, and so now we're thinking of moving it into one of the main research lines in the lab. But first, let me introduce myself. My name is Erill, and I'm originally from Barcelona, although now I'm an associate professor at University of Maryland. And I'm a computational biologist / bioinformatician, or whatever you want to call it. And as you know, bioinformatics, we're very fond of omics words. So there's so many that are hard to count. I think the last count on Wikipedia was like 70. And I decided to create my own a few years ago. So I created the researchsome. And the researchsome is just a way of depicting my research domain. And as you can see, I'm mostly working with bacteria and with transcriptional regulation. And that's basically the whole focus of the lab. We work a lot on modeling transcription factor binding site interaction, so the process of proteins recognizing DNA. We work on developing comparative genomics tool sets and applying them for discovery on transcriptional networks in bacteria. And we also work on performing evolutionary simulations and doing in silico evolution to see how evolution networks, what design principles evolutionary networks should follow. But today I'd like to talk about two specific aspects of my research. One is stress responses, in which I have worked for many years. And the other one is one of the trendiest omics words these days, which is metagenomics. And so there are essentially two buzzwords for my presentation today. One is regulon, and the other one is the metagenome. And I'm assuming from the faces I see that everybody knows what a metagenome is, so I'm going to introduce what a metagenome is and probably make some inaccurate statements, so forgive me. But first with the regulon. So a regulon, by definition, is a set of genes that are regulated at the transcriptional level by a transcription factor, which is essentially a protein that regulates them. And the transcription factor typically reacts to some either internal or external stimulus. And in doing so, what it does is it activates a network of genes that basically coordinates a cellular response against to something. And that something could be, for instance, a change in pH, and the transcription factor would react to the change in pH and then activate a set of genes that would, for instance, if it's a pH change, then you would expect to see proton pumps being activated or changes to the cell wall or increasing the rate of production of chaperones so that proteins fall correctly in spite of the change in pH. And so the neat thing about regulons is that they are basic elements in these huge hairy balls that you see that are called the global transcriptional regulatory network. And these are basic subunits of the regulatory network. And again, they are defined by specific binding of a transcription factor that's able to respond to stimulus to the promoter region of target genes or operons. And in order to do this, so once the transcription factor binds to the promoter region of a target gene, it can either help or hinder RNA polymerase in initiating transcription, and therefore you can activate or repress a gene. And the cool thing is how transcription factors know where to bind in the genome. So the genome, even for bacteria, the genome is quite large. 4, 6, 10 million base pairs. And so what transcription factors do is essentially search, locate, and bind small, short sequences in the genome looking for a specific pattern that's called a transcription factor binding motif. And so that pretty much is, in a nutshell, what a regulon is. As for the metagenome, so in traditional genomics, we typically isolate a pure culture of some bacterial species, and then we culture it until we get a lot of DNA, and then we lyse it and sequence it. In metagenomics, what we do is we basically go into whatever, the environment, which can be strange places for some people, but still the environment. Can be the human gut. And we go there, we get a sample, and we lyse this using very different techniques, but essentially we apply high throughput sequencing, and we get what essentially is a multi-species heterogeneous collection of reads from a natural environment. And that has some good, bad, and ugly properties. The good properties that we get to see lots of unculturable species. There's some debate as to what unculturable means, but it basically means hard enough for a lazy biology to figure out how to culture. So there are things that are not easy to culture and therefore are uncultured. So you get to see these guys, which represent by some estimates 90% of the total bacterial diversity out there. We also get to see a lot of diversity in terms of species, substrains that we would never see in standard genomics approaches. And we get to see also not just the diversity there, but we can quantify that. So we can not just see what's there, but what are the proportions of the things that are there. The bad news is that we start with little material. We don't purify pretty much, so we just extract the DNA. So it's hard to get good coverage. You need to spend a lot of money, much less these days, but still money. You need to deal with polymorphism, which you don't typically have in a clonal monoculture, which can be something, can be good, but it's typically bad. And then we have all sorts of stuff going on. We have multiple species. Every species had its own paradigm for low complexity regions and so on and so forth. So the bad news for bioinformaticians is that we get a system that's so heterogeneous that in practice is very hard to train proper models for things like assembly, gene calling, and all that stuff. So what used to be problems that were pretty much solved in standard genomics now become hard problems again. And in fact, there are some people that claim that metagenomics fits so well the paradigm of bioinformatics because we have lots of data, it's extremely noisy, and it's increasingly cheap and abundant, that there are some people that claim that metagenomics was invented by a conspiracy of bioinformaticians who feared they would go out of a job in the next 10 years because genome sequencing would get fully automated, so they would be, not needed anymore. So they decided to do something new and more complicated, so they would keep their jobs for at least 10 years more. But the nice thing is that at least some of those bioinformaticians start to work hard on these new problems. And these days, when you get a metagenome, what you typically get, once it's post-processed, there are now good assembly pipelines and there are good gene calling methods for metagenomes. So what you get is typically these days assembled contigs or scaffolds with predicted genes and oftentimes to functionally annotated genes. So it's not such a bad data set to work with. But the problem is, what do we do with the data? And so to put it in Sidney Brenner's words, how do we escape from the low input, high throughput, no output science stigma, right? So how do we move from getting all this data and then generating some useful knowledge with it, which at some point supposedly it's supposed to be the goal of the whole thing. And so the standard answer to that has been, to date, to analyze metabolic pathways. And the way this is done is you take your data and you basically throw it against a terminal setup, a terminal knowledge base. So you map it against known pathways and you map it against known taxonomies. And then basically what you get to see is what genes are doing what in the metagenome and who these genes come from, right? So you get a picture, you can get a picture of a shared metabolic pathway and see exactly who's contributing different genes to that pathway. And this leads to very interesting questions. So Jeffrey Gordon's group at University of Washington, St. Louis, and Rob Knight, which are both featured here, have been working a lot on this kind of things, and they have been asking questions such as, how does your microbiome impact on your health, obesity, whatnot, and apparently getting conflicting results depending on their methodologies. But who cares about the results once the publication's out? But one of the things that interested me when I started reading these papers was that they were starting to ask questions that were linked to evolutionary biology, which also has always been my passion. And so they were asking questions like, okay, so is the microbiome in mammalian species, is it dictated by diet, so convergent evolution to diet, or is it just a question of radiation of the mammalian tree of life? And so the answer they got here was basically that it was mostly diet, and they actually saw cool things, right? So they saw that biosynthetic pathways for amino acids were enriched in herbivore microbiomes, and biosynthetic degradation pathways were enriched in carnivores. So, which means that much like our genomes, our microbiomes are a mirror of our culinary preferences. But so this got me thinking, and the problem I had with this whole workflow is that because the knowledge base is terminal and you're just mapping against it, you can basically do two things. You can cluster, you can filter, and that's it. So the potential for discovery of new things is low because your knowledge base is static. It's predefined. So it's hard to actually do new discoveries using this type of approach. And so my field is transcriptional regulation. So I started thinking these are metabolic networks. What about regulatory networks? What about regulons? Could we use metagenomic data to get to know something about regulatory networks? And I started thinking of an alternative workflow that we could use. And so here's the deal. The idea here is that our prior information are a transcription factor and its regulon in the form of what genes it's regulating. And we can distill that even further into what is the specific binding motif that transcription factor likes to identify, likes to look for when it's trying to locate its binding sites in the genome. And this is all our prior knowledge. So we have some prior knowledge, we squeeze it into a binding motif, which we can represent as a position-specific weight matrix, and I won't go there, but you can just score sequences and try to see how well they match this pattern that you see here represented as a logo. And then you can throw this against the metagenome, search the whole metagenome, and what you get back is some estimate of what the regulatory network looks like in the metagenome. And so that was the idea. I'm not very smart, so it was a dumb idea. And so if these were feasible, then you can start asking interesting questions. You can start asking questions like, do regulatory networks, do regulons evolve convergently to adapt to a habitat? Or are they basically following the radiation of bacteria? And so bacteria from the same clade have the same type of regulon, regardless of the habitat they are in. But of course, before we could start asking this type of comparative questions, the first thing we had to prove was that we could actually do this dumb thing and get something meaningful here as the result. So that we could take a known regulon, distill it into a binding motif, search this whole pile of metagenomic data, and get back something that looked like a regulon and see if it matched our original regulon, and therefore we could claim that with some confidence we could reconstruct the original regulon. And of course, if you were able to do this, there are advantages to using metagenomic data. Again, we get to see real bacterial populations. We also get to see all the unculturable or uncultured elements, mobile genetic elements like phages, transposons, all that stuff that's floating around and typically not sequenced in a normal sequencing, genome sequencing project. And we get to see a lot of variability in terms of polymorphism and strains and substrains that's not visible in genomic approaches. So those are the good news. The bad news is that searching for a small signal, like in this case, 18 base pairs long, within a data set that's 8 giga base pairs long, it's going to result in tons of false positives. So it's a noisy search process, and we knew that beforehand. And so it was not clear that we would accomplish anything by searching that thing. And secondly, once you get the search results back, it was not clear also how we should proceed into integrating those results, performing some enrichment measures, and analyzing the results. So the goals for the project when it started was to show that indeed we were able to get metagenome information, get basic prior information on a regulon, and using both reconstruct or extract knowledge on the regulon, on the metagenome. So get information on the regulatory system from both the prior knowledge and the metagenomic data. And we decided to do so just by studying one regulatory network in its natural setting. So having decided this, we only had to choose two things. One was a regulon or regulatory network to analyze, and the other thing was a metagenome to analyze it on. And so the first answer was the bacterial SOS response, and that to me was a no-brainer because I've been working on this specific response for pretty much 10 years. So I know it well, so it was an easy target, low-hanging fruit for me. So what is the SOS response? It's a response against DNA damage, hence the name SOS. Again, I had worked on this response specifically, so I knew it well. It's also a good pick because it's widespread in bacteria. It's been well characterized in a bunch of different phyla. And in terms of setup, it's actually quite simple. It's an approximation to a two-component system. There are two main proteins involved, REC-A, which acts as a surrogate sensor, and LEX-A, which is the master repressor of the system. And the way it works is LEX-A is a repressor, so it works as it's a dimer, sometimes forms a tetramer, but typically a dimer. Locates its binding sites called SOS boxes upstream of the genes it regulates and represses them very tightly. It's one of the strongest bacterial repressors. And so when there is DNA damage, what happens is bacteria are always replicating, and they're chromosome. And at some point when there is a lesion in the DNA polymerase is picky. It will stop, detach, and leave this mess with an open fork stalled, which generates single-stranded DNA fragments. This is very dangerous for bacteria because endonucleases will come in, eat the whole thing up. So REC-A binds single-stranded DNA, helps protect the replication fork. But it also, once it becomes activated by binding single-stranded DNA, it's able to promote the self-catalytic cleavage of LEX-A. Essentially, LEX-A repressor performs a harakiri, kills itself. And once it does that, it's not able to bind to those sites in the promoter regions of the genes it regulates. And once that happens, all the genes that used to be tightly repressed suddenly become expressed at constitutive levels. And what gets expressed, what gets activated is, for the most part, three functions. DNA damage repair, so a bunch of enzymes to repair the damage. Cell division inhibitors, so that you don't split your cell before you've managed to complete replicating your chromosome. And translation synthesis polymerases, which are less picky than the normal polymerase and will complete replication of the chromosome even if there is still DNA damage left. And in doing so, they will typically create mutations, so it's a sort of a mutagenic system. So that's the classical textbook of the SOS response, but there's a lot more to the SOS response that made it clinically relevant. So in the last few years, it has become pretty well established that the SOS response does not only respond to classical DNA damaging agents, it also responds to pretty much everything that can eventually cause DNA damage, and that includes pretty much every single antibiotic. And among the genes that it activates, the SOS response has been shown to mobilize a ton of different mobile genetic elements, from pathogenicity islands to bacteriophages, recombination of antibiotic-resistant cassettes in integrons, and you name it, transposons, everything jumps, basically. The SOS response is a signal saying this cell is not functioning properly, there's DNA damage, leave now. And when these guys leave, of course, they leave with whatever they carry, and in many cases, that's antibiotic resistance genes. So what you get, in essence, is a feedback loop in which you can treat with antibiotics and promote dispersal of antibiotic-resistant genes, which is a bad idea. So that's what's basically put the SOS response in the limelight in the last 10 years or so. And beyond that, again, I'm prone to be interested in evolutionary questions. The SOS response is weird in several aspects, but so it's widespread in bacteria, but it's absolutely absent from some groups of bacteria, including the Bacteroides, which have relevance for metagenomes and human guts. And the other thing is that in contrast to most transcription factors, which tend to look for the same pattern wherever they are, whether they are in gram-positive bacteria or they are in gram-negative bacteria, they always look for the binding site, the binding motif that they look for is conserved. Let's say it has changed its binding motif like crazy in many different groups. For instance, in E. coli and all the gamma protobacteria, it looks for a palindrome that looks like this. And in gram-positive bacteria, it looks for a palindrome that looks like that, and they have nothing like pears and apples. So the interesting part is that in each one of these groups, the Lexay binding motif is monophyletic, meaning that this is the way the gram-positive Lexay binding motif looks like, and it looks like this in every single gram-positive bacterium. And that was a cool thing for the SOS response in terms of the process that we were trying to do, because it allowed us to filter groups of bacteria, and we could narrow our search to specific subgroup of bacteria just by determining which binding motif we were looking for. So, and in this case, we chose the gram-positive bacteria as our target group. And that's it for the SOS response. As for the Metagenomics Project, we chose the human microbiome for obvious reasons, because it has clinical relevance. But also because when we started this project a few years ago, there were already two main initiatives going after the human microbiome. And the data from one of them, which was the MetaHit project, was very good. And in particular, for us, it was good because it had large context. And that meant that we not only had small segments of reads with a gene in it, but we also had genes and the upstream regions in which regulation takes place, which is where we wanted to look at. So we chose the SOS response and the human microbiome as our target, the human gut microbiome as our target. The other thing about the human gut microbiome is that it's dominated by Firmicutes, which are gram-positive bacteria, and Bacteroidetes. And so we had a natural control. We knew that here there should not be SOS response at all because it's not there. And it's always good to have a natural control for sanity purposes. Whereas for the Firmicutes, we knew there is a very well-defined Lexay binding motif for that particular subgroup, so it was a very clear cut. So that was it. And so again, using my amazing brain, I devised a super difficult bioinformatics pipeline, which I'm going to summarize in a couple of slides. The first thing we needed to do was get data. We got a comprehensive description of the Lexay binding motif in gram-positive bacteria We did that using our own database called CollectTF, which is a database that we have on transcription factor binding sites in bacteria. It has data on more than 150 transcription factors over probably more than 90 bacterial species. And it's growing, and it's an open access thing in which you can submit your own data if you want to, and it gets pushed to NCBI, so it gets a lot of visibility. So basically, we collected data from all the known bacterial species, gram-positive bacterial species with known binding motifs for Lexay. This is how they look. This is how they look when integrated. And this was basically our target motif to search for in the metagenome data. We also collected reference genomes from both MetaHit and the Human Microbiome Jumpstart project to get a reference panel for classifying taxonomically the genes that we're finding. And we also mined the literature to get full descriptions, or as full a description, global description of what the SOS response is in 8 different bacterial classes, totaling 18 different species, and with nearly 300 genes described as being regulated by the SOS response. So with all this data in hand, all that remained was start searching. So we searched the whole metagenome. That's 8.1 gigabase pairs, I think, both strands. That's a lot of time. But basically what you do is you take your motif and you start looking for things that look like it using a position-specific weight matrix. We discard things that were 1.5 standard deviations below the mean for the reference collection so as to filter out without some noise. But essentially, we just went searching and annotating every single site above that cutoff that we could find. Once we did that, we predicted operons using a very simple dumb algorithm, fast algorithm for operon prediction. We were not interested in accuracy, but in yield. And once we had binding sites and operons, what we did was associate binding sites and operons, and we did that just distance-based. So we associated the binding site to the operon based on the distance from the binding site to the first gene in the operon. And then for every single gene that we had associated to site, we blasted it against the reference genome library to get both a functional description, but for the most part, taxonomical description. So where did this gene come from? What's the closest relative in reference genomes for this gene? So that we could then apply some taxonomic filtering. And then we blasted against the COG reference database so that we could get clusters of orthologous genes, so functional annotation on the genes. And that was it. Took a couple of days of compute time, and that was it. And so basically, this is the data we had for analysis. We had predicted sites, predicted operons, sites associated with operons, and for each gene within each operon, we had both information on what the closest bacterial species was for that gene from the blast hit, and also information on what the COG was for that gene, if there was a matching COG. And so all that remained was start scratching our heads and analyzing the data. And so the first thing we did was sort of a sanity check. The search produced like 500,000 putative Lexay binding sites, which is a lot. And the first thing we did was check whether there was enrichment in Lexay binding sites in the promoter regions, which is where you would expect or at least hope that they would be enriched. And that's actually what we saw. This is coding region and this upstream region from about plus 50 to minus 500. You see a clear enrichment in binding sites in the presence of detected binding sites. And the enrichment becomes more and more pronounced as you increase the cutoff for what you consider it's a putative binding site. So as you make it more stringent, you see a much more clear response. So we then decided to quantize how much statistical power there was here. We did a permutation analysis in which you basically take your binding motif here. Where is my binding motif? And you start flipping columns randomly. And then you search, again, those regions, and you look at the distribution of scores. And if you're lucky, you're going to see something like this. The blue line is the original motif. This is a histogram. And the red lines are permuted matrices. So essentially, if you search with a true motif, which supposedly is preserved by evolution because it has a function, you see at high score levels, you see more sites than you would expect by chance. So, okay, so we thought we're on the right track. We're doing something correctly or more or less correctly. But there still was too much data to process with 500,000 putative lexi binding sites. So we decided to restrict and narrow our analysis. And we did that with a two-pronged approach. So we first applied a distance-based filtering. We just considered sites that were upstream of operons, of the first gene in an operon. And we also decided to consider only sites that were associated with operons in which the genes map to gram-positive bacteria. So we had the binding motif that was specific for gram-positive bacteria. And the genes had to belong also to gram-positive bacteria. And applying these two-tiered filtering approach, what we got was a hefty reduction in the number of putative sites that we found. So we got down to 43,000, which is much more tractable. And we also found that taxonomy-based filtering approaches are essential to this kind of work in which you're dealing with heterogeneous data sets, because if you search enough data, essentially you're doing a huge multiple hypothesis testing. So you're going to find things that are highly scoring, statistically significant anywhere. And so our control, which were the Bacteroides, which don't have the SOS response, and therefore we should not expect to see lexi binding sites in the promoter regions of these guys. But of course, if you look enough, you'll find lexi binding guys in the promoter region. So using the taxonomic filtering was a great aid to focus the analysis and enhance it. So, okay, so we had found that there was positional enrichment. We said, okay, what about functional enrichment? And our second sanity check was to look at COG categories. So COGs are a cluster of orthologous genes that are defined by sharing typically sequence features that and clustering them into several genes that have similar domains and therefore supposedly have similar function. And these COGs are further clustered into categories that have broad functions. So like replication or DNA repair. And so what we did was we looking now just upstream of genes, at the binding sites that are upstream of genes. What we looked was at the enrichment in COG categories as a function of the scores that we were using as a cutoff for the sites. And as you can see, as we increased the score that we were using to define what a functional site was, we got closer to the green line. The green line is the reference, those 18 reference SOS responses that we had mined from the literature. Those are the COG categories for those guys. And so as you increase the cutoff and you become more selective, you get closer and closer to the green lines, which were our reference. That happens for everything but D, which is cell control. So we saw everything we wanted to see. We saw L, which is DNA repair, obviously. T, which is transduction, and what's the other one that's enriched? K, which is transcription. So those are three expected things. We also expected to see cell cycle control because the SOS response is known for controlling cell replication, stopping it before you have repaired the damage. And what we saw was that all the genes from gram-positive bacteria that have been later described as being cell division inhibitors, when the COG database was created in 2002, I think, 01 or 02, they had not been described, so they were all put here in the unknown categories, and that's why we were not seeing the enrichment that we hope to see here. That small green line, we never got that. But apart from that, everything else was kosher, so we decided that we'd move on and look at individual cogs and try to see if we could make calls on whether a cog was a putative member of the SOS response or not. And that's when we started running into trouble, because it was not clear how to define what was an enrichment measure for SOS cogs, right? So we're trying to identify cogs that belong to the SOS response in gram-positive bacteria in the human gut microbiome. And so our goal was to find something that was able to identify members of the regulon and that would capture as much of the knowledge that we had on the response, hopefully. And so we started looking at different techniques, standard techniques for enrichment, the ratio between high-scoring sites and low-scoring sites, and stuff like that. And it didn't work out nicely because the metagenomic data is very heterogeneous, and so it's not very clear-cut what you should be expecting. And we finally decided to look at genomes, and we looked at the two genes that are always, pretty much always, regulated by LEX-A. We looked at 300 gram-positive genomes, looking for these two genes and looking at the score distribution of LEX-A binding sites upstream of these two genes. And what we found was that it was pretty much normally distributed. So the scores of LEX-A binding sites upstream of these two genes were normally distributed with a mean at around 16 bits. And that meant that the cumulative distribution was approximately linear in the 12 to 20 bit range. And so what we did at that point was to define our version of what should be a prototypical SOS cog. We defined that a cog would be a member of the SOS meta-regulon if it had a high linear coefficient of determination for the cumulative distribution in this range, and if it had at least one site scoring above the average, and at least 10 sites in that range, so that there was enough data to make a call. And that was basically a hack. I'm not happy with it, but it worked. So I'm going to shut up and leave it there and hope that the biostatisticians in the room don't ask questions. So we basically were using the linear coefficient of determination as a proxy for normality, which is half-assed at best. But anyway, it worked. We used this. We ranked all the cogs using this metric. And what we found was that we could identify with high confidence the canonical SOS response. So we could, in green, you have known SOS cogs, including the master, the sensor, the master regulator, LEX-A, and so on and so forth. This is their linear coefficient of determination. And so we could detect repair functions, excision repair, recombination. We're also able to look at, because we're looking at the metagenome, and we have data on abundance of sites in front of cogs. So we could not only tell what was there, but how much it was there. So how often did you see, how many sites did we find upstream of one type of cog versus another type of cog? And what we found was that traditionally the SOS response has been seen as a DNA repair mechanism. That's been the textbook paradigm forever. And translation synthesis has always been seen as something as an add-on to the response. And what we found was basically the opposite. So apart from the master regulator, which is always regulated and always has sites upstream of it, the other second cog that had most frequently sites upstream of it was the cog belonging to translation synthesis polymerases. And that's in agreement with some results by my group and by other groups suggesting that translation synthesis is the primary function of the SOS response and that the DNA repair came after. And there is some evidence to support that. And in between all these known cogs, we were able to identify 3 cogs that had never been reported previously as being linked to the SOS response. Some of them, 2 of them, are linked to cell division and control, and therefore make sense in terms of what the SOS response is. And actually, one of these guys had already been reported as being induced by antibiotics, but nobody knew the mechanism. And these restriction endonuclease that I think in Streptococcus suis had been reported as being involved in virulence and resistance to phage invasion, which again makes sense in the context of the SOS response. So we identified 3 new putative SOS cogs. And so the last thing that we wanted to check was whether we could leverage metagenomics data to look for specific targets that either look promising, look cool. They came from things that might be unculturable, like phages, stuff like that. And so what we did was a sort of a targeted analysis of genes. So we identified, using the permutation analysis, we identified what were high confidence, high scoring binding sites for, let's say, in the metagenomic data, and then we cherry-picked some of them. And we looked at whether we use electromobility shift assays to see whether purified Bacillus subtilis, which is a gram-positive bacterium, Bacillus subtilis lexate protein would bind there or not. And so basically, we validated binding of lexate to a toxin-antitoxin system, which has already been linked to persistence phenotypes and in different bacteria, and has already been shown to be regulated by the SOS response, I think, in E. coli. We also saw that some phage integrases are regulated by the SOS response, and this kind of makes sense. Our group had shown in 2009 that integron integrases are regulated by the SOS response. Integrons are basically gene swappers that can take in or out gene cassettes and share them. And also we validated one of the cogs that we had shown that were enriched in our previous analysis. So the cell wall biogenesis cog, we validated one of the sites that were associated with MRAZ, which is a gene involved in cell wall biogenesis. And again, it proved we could show that it was bound by lexate in vitro. And so that sort of shows evidence of convergent regulation of cell division in gram-positive bacteria. There is a bunch of different genes that control cell division in gram-positive bacteria, and different species have different genes, but they are all regulated by the SOS response. So identifying new members of this category sort of made sense too. And that's basically what we did. And some sort of conclusion, we were able to prove that indeed you can actually go and do this dumb thing of taking a motif, throwing it against the metagenome, and fishing out things that make sense and that match our prior knowledge on what the metagenome is. So it's doable. We sort of established a methodology which is not perfect, but which we can improve upon. What we saw was consistent with the known SOS responses, putting an emphasis on mutagenesis, which I like because it was my theory. And we also identified things that are involved in virulence and so on as being linked to the SOS response. And so again, our future direction with this, the initial idea was to be able to do this automatically, get one regulon, one metagenome, one regulon, another metagenome, compare them, analyze and compare, and then ask questions as, is the habitat determining what the regulon looks like, or is it the composition of bacteria and the different metagenomes dictating what the regulon looks like? And so that's what we're working on now. One of the things we're working on is trying to automate this inference. So what we did for this case was cheating, right? We knew the answer before we started. So, holy cow. Sorry about that. So we knew the answer before we started. We knew that we knew exactly what cogs, or at least some of them, we should expect to see enriched. we should expect to see enriched. And therefore, if you look at the distribution of scores in upstream regions for the cogs that we know, the red ones are the ones that we know should be SOS cogs, the canonical. You see this very nice departure from a normal unimodal model to a mixture model in which you have the expected distribution and then selection going on here. Whereas if you pick just random cogs, that disappears, or if you just pick you don't filter for a specific bacterial group, you just put them all together with the bacteroides and so on, that disappears too. And so that's very nice if you know the answer, and that's what we use to make our calls on what cogs should be enriched. And that's how we were able to come up with this ranking system. But if you don't know the answer, this is how the whole distribution looks. And this thing here is there. And so if you don't know that's there, the question is how do you find it, right? And so what we're working on now, and I won't go into details, but is generating an expectation maximization algorithm to sort of try to automatically isolate cogs in taxonomic groups that are enriched in binding sites for the transition factor. So the idea is whenever you have a transition factor, it may exist only in a subset of species. It does not have to be in all the species that you're analyzing, so not all the species should be in. And you know for a fact that it's only going to be targeting a subset of the cogs. So one thing you can do is if you define a statistical test for a mixer model, which is what we see here, if you define such a test and use it as a likelihood function, you can plug that into an expectation maximization algorithm in which you basically say, okay, given this fixed distribution of cogs, let's revise my belief that each of these taxonomic groups is enriched in sites for so has a small lump at the right tail of the distribution or not. And once you revise serially all your taxonomic groups, you fix the taxonomic groups and you revise the cogs and you iterate this process as a standard expectation maximization algorithm. And the hope is that it will be able to isolate. So I have 2 students working on that right now. I'm hoping to get an email at some point saying that it works. But that's one of the directions we're working in. We're also automating the pipeline and trying to make it, standardizing it so that if we're comparing metagenomes, we do exactly the same protocol on all metagenomes. And that was all for this talk. I'd like to thank Joe Cornish, who was an undergrad in the lab at the time, he's a postback at NIH now, and he did most of the in silico work, and Neo Sanchez Alberola, who did the experimental shift assays to validate sites, and then collaborators at both the Wolf Lab and the Barber Lab, and of course funding both from UMBC, my home university, and the NSF. And I'll take any questions.